will be touching upon there's a small. Yeah. Uh, and what do we do is we work in the areas which I shall be highlighting, and we also would like to
Now I would like to call Dr. Himanshu to come on the stage and speak about our outreach program.
To bridge the gap between the cutting edge research and ground level impact. Each page of this document tells a story of curiosity kindled, of lives touched, to bridge the gap shared between the cutting edge research and ground level empower communities. Each page of this document scientific excellence story and compassion of curiosity to animal health and of lives touched. This document serve between as both cutting edge research and ground level that science, when extended beyond walls, has the power to uplift, inspire, and transform of lives touched. Now, I humbly request our esteemed guest for today's event, Dr. Vijay Kumar Saraswat sir, our respected director madam, and Professor Redana sir, to officially unveil this document. Please, sir, please. Now, I humbly request our esteemed guest for today's event, Dr. Vijay Kumar Saraswat sir, our respected director madam, and Professor Redana sir, to officially unveil this document. Please, sir, please. Now, I humbly request our esteemed guest for today's event, Dr. Vijay Kumar Saraswat sir, our respected director madam, and Professor Redana sir, to officially unveil this document. Please, sir, please. Now, I humbly request our esteemed guest for today's event, Dr. Vijay Kumar Saraswat sir, our respected director madam, and Professor Redana sir, to officially unveil this document. Please, sir, please. Now, I humbly request our esteemed guest for today's event, Dr. Vijay Kumar Saraswat sir, our respected director madam, and Professor Redana sir, to officially unveil this document. Please, sir, please. Now, I humbly request our respected director madam, and Professor Redana sir, to officially unveil this document. Please, sir, please. Now, I humbly request our respected director madam. Thank you, sir. Thank you, ma'am. Now I invite Dr. Sonu Gandhi to proceed further with the program. Thank you. Thank you, sir. Thank you, ma'am.
Thank you, Himanshu.
Now I invite
So
Dr.
it is my
Sonu
pleasure
Gandhi
to
proceed
introduce
further with the
Sir
program.
Dr. Vijay Kumar
Thank you.
Saraswat on the occasion of 14th Foundation Day of NIAB. Dr. Saraswat is not just a scientist or a policy maker. He is a trailblazer who helped to shape modern India's defense and technological strength. Dr. Vijay is an eminent scientist who formerly served as the chief of the DRDO and scientific advisor to the Indian Minister of Defense. Dr. Saraswat is presently a member of NITI Aayog, and he was also former Chancellor of JNU and former President of Sri Chitra Tirunal Institute for Medical Sciences and Technology, Trivandrum. Dr. Saraswat is the key scientist in the development of the Prithvi missile and its induction in the Indian Armed Forces. He is a fellow of National Academy of Engineering, Aeronautical Society of India, Astronautical Society of India, and Institution of of Engineers. He is a member of Governing Council of SAMI and member of Board of Research of AICTE, CSIR Labs and Board of Studies of Osmania University. He is the chairman of Combustion Institute, Indian Section and Aeronautical Society of India, Hyderabad branch. Dr. Saraswat is the recipient of DRDO Scientist of the Year Award, National Aeronautical Prize, DRDO Technology Transfer Award, Aryabhatta Award, Performance Excellent Award, and many more. For his outstanding contributions to the nation, he has been conferred with the most prestigious Padma Shri in 1998 and Padma Bhushan in 2013. In January 2010 and December 2012, he was awarded with the honorary doctorate from Sardar Vallabhbhai National Institute of Technology, Surat, SRM Institute of Science and Technology, Chennai, and JNTU, Hyderabad, respectively. With this, I would like to invite Sir and putting a very warm welcome. National Institute of Technology, invite you to the stage. Thank you. With this, I would like to invite Sir and putting a very warm welcome. National Institute of Technology, invite you to the stage. Thank you. With this, I would like to invite Sir and putting a very warm welcome. National Institute of Technology, invite you to the stage. Thank you. With this, I would like to invite Sir and putting a very warm welcome. National Institute of Technology, invite you to the stage. Thank you. A very warm welcome.
Namaskar, good afternoon. Dr. Taru Sharma, Dr. Adanya, Dr. Kashmurti, and all the esteemed invitees for this Foundation Day celebration. The scientists of National Institute of Biotechnology, Dr. Taru Sharma, Dr. students, Dr. Kashmurti, and all the other and all the members of the NIAB for this Foundation At the onset, let me congratulate you for your 14th Foundation Day. Many happy returns of the day. Students, I have seen the growth of this organization members of the under the leadership of Dr. At the onset, let me it started in a small way at CRO Institute. In those days, we had also just started I have seen the growth of this organization and Dr. CRO was alive and used to be part of our deliberations process. I think from that and the story which was narrated by Dr. Tarak really shows where the institution has grown. And I really appreciate the wonderful work which has been done by all of you. From my hearty congratulations and you should keep the spirit of this research going. In your adolescent years, because you are now 40, I really appreciate the wonderful 16 and then so I think so that is the kind of growth this organization should have, and my best wishes on this day. I'm not a biologist, I'm not a biotechnologist, I'm a missile scientist, I'm an aeronautical, so that is the kind of mechanical engineers or electronics you could have, whatever, and my best wishes are forgotten. What is my daily discipline in which I'm not a biotech work on, because by interacting with people like you for the last so many decades, I think something or the other of every discipline has and that's why I'm very comfortable because when coming here, by interacting with people, my talk is basically not new to you. I think you are working in this area. I'm only going to highlight some of the aspects which I felt are important, but for you it may be a repetition also. My talk is basically I'm going to talk to you biotechnology in an era of biological revolution. All of us know that we have had different decades and we have different basically innovation. In the sixth wave of innovation, biology has found, biotechnology has found its name. We all must have heard about synthetic biology and green chemistry and biotechnology as the basic pillars of the sixth wave of innovation which is currently on. So obviously in that sense, we all must have you have synthetic to play because it is now the intersection of all other disciplines with biology, which is what is going to lead to the new discoveries, new innovations. So obviously in that because we are all looking towards going as close as possible to the nature. But unfortunately, our understanding of the nature has been very, very low. And I think biology is one which is going to help us understand. We are all looking towards the human aspects, the nature aspects, and so on. But unfortunately, our understanding of the nature has been now the biological revolution of 21st century, because it is not just digital. People talk nowadays digital in a big way, but unfortunately, it is biological. Science is now engineering life with the precision considered for foods and circuits. It is like not just digital. So life is becoming nowadays. That's why Hyderabad is a place where I think we are very lucky that we have the highest number of institutes where bio life sciences are being talked about. Life is becoming research. That's why for India, with its vast biodiversity, livestock wealth and traditional knowledge systems, this shift is not just an opportunity, it is a national inflection point which is taking place. Among that, animal biotechnology sits at the heart of this evolution, which is enabling sustainable protein systems, reproductive gains, disease resistance, and genomic conservation. Among that, animal biotechnology sits at now how we have reached and where we have reached. Our indigenous breeds, once seen as a liability, disease yield rates are now genetic reservoirs, adapted to the climate, disease, and marginal geographies. Globally, nations are turning bioassets into geopolitical space. I don't know how many of you have realized that point. From vaccine diplomacy to gene edited trade flows. So it's becoming a consumer globally. Nations are turning bioassets into India must not remain a consumer of biotech revolutions, it must shape them now. From vaccine the challenge before us is clear to convert our genetic wealth into strategic advantage and to do so with speed, ethics, and scale. This part is very well known to biotechnology, and in that now we are talking of animal biotechnology covering livestock, poultry, companion animals, fish, insects, laboratory animals, and so on. This part is very well we are talking of creating animals with one or more genes introduced by human intervention. And that is what I saw, the kind of research you people are doing for insects. Employing gene knockout technologies specifically inactivated genes and making virtually identical animals using somatic cells, nuclear transfer, or cloning. This is what we all are looking at. How this world is taking place and how it has happened from the days when we had nuclear transfer or cloning. And wild animals were domesticated, so we had domestication, which led to the tame, manageable and now then we got into selective breeding by doing cumulative incremental genetic improvements in the reproduction traits. So that resulted in specialized breeds with improved production and potentially higher resistance levels. What we are seeing. Then you people started doing genomic selection. That means you expedited genetic improvements of multiple traits. That led to breeds with further improvement in many performance traits. Then now you are subjecting them to new biotechnology methods, which is a step change in performance rates and disease resistance. But beyond that, now the new technologies which have happened, like genome editing, transgenesis, technology to introduce new genetic variations, new technologies to generate genome edited animals, primordial germ cells technology, cloning, zygote, microinjection, and so on, transgenesis. So basically, we have integrated new breeding technologies into commercial breeding programs and production, which has led to the livestock 2.0 germ cells. We are talking about highly productive and disease-resistant animals for a range of environments. That is how the progress has taken place. And this is how you people are now growing. Modern biotechnology is evolved from traditional domestication and selective breeding to precision breeding, including marker-resistant selection and gene. And this is how biofarming is now growing and sustainable development for food security and climate resistance or climate resilience, I must say. Stem cells and marker resistance, also a lot of work being done here also. And improve diagnosis and diagnostics. So that is all is a part of your research. Biofarming, this word was also used by Dr. Saraswat in her presentation, where we are looking at transgenic livestock, which can basically bioengineering on the farm for production of therapeutic biofarming, where it can be used as bioreactors, livestock, to produce therapeutic proteins and pharmaceutical in their milk or other bodily fluids, offering a cost-effective method for drug development. Genetically engineered animals can be used for producing specific enzymes for industrial processes. This diagram very clearly indicates how offering the DNA of a goat and the human gene can be combined to create an egg transplanted into a goat, and from there how you can get the different diagram very clearly indicates. So this is a kind of biofarming is going on. Then you have the science is your area, which you are doing day in and day out now, genetic improvement, where use of CRISPR, Cas mediated gene editing system for generating disease resistant. Then you have herbs, which is a homologation of three items like host immune system, host genome, and the pathogens, which leads to the application of genomics and identification of candidate genes to look for that for disease resistance. Again, apply the CRISPR or Cas9 genome editing for the target gene, and finally you will end up with disease resistant animals. The cycle is also shown on the right side. I think you know. Again, apply the CRISPR or Cas9 genome. Biotechnology has been responsible for climate resilience animals. Animal biotechnology offers strategic solutions to mitigate and adapt to the impacts of climate change in the livestock sector. For example, climate resistant breeds, which are heat tolerant, disease resistant livestock. Then you have major problems with the livestock in terms of methane. So methane mitigation, and that is how you optimize the gut microbes, ensuring that the methane is reduced. Then you have, of course, sustainable feed and resource use, where nutrient efficient animals are done, and early disease detection and management, where minimize disease outbreaks and lower environmental footprint. That is another one where precision breeding is involved. So animal biotechnology offers strategic solutions for climate resilience, climate change mitigation, and adaptation in the livestock sector. Innovations such as low emission breeds, enhanced feed efficiency, and disease resilience are critical for reducing the environmental footprint of animal agriculture. Methane is one of the major problems, as all of us know, and so methane mitigation strategies are there, which are basically related to animal and feed management. While you can do methane processing to reduce that, you can do certain kind of genetic selection. You can also do improve the health of the animal by various methods, and improving the pasture management where they are going to graze. So that is the one area where genetic selection, increasing their feed levels, increasing forage quality, and optimizing the temperature. Similarly, diet formulations like use byproducts, decreasing the forage to concentrate ratios, use minerals and salts and oils and fats, use oil seeds, increasing protein or turning failures, forages or urea. Decreasing, of course, human manipulation is also use minerals and salts and oils and fats. One of the major factors which has been responsible for India to become number one turning failures is our milk yield. The milk we are producing human manipulation. The process is nutrigenomic approaches where enhancing the feed digestibility to increase the tailoring the feed for altering what we call it the microbiome. The process and that leads to the higher yield. Milk production reached almost 230 million tons in year 24, which was early 230 million tons in 2023. Nearly 4% year-on-year growth, maintaining India's position as the world's largest milk producer. So that is what, which was early 230 million tons. You already talked about one nation, one health, and so on. But animal biotechnology for advancement in the human health is very important. So that is what. One of the things which is possible is the disease model. Already talked about. So transgenic animals serve as a crucial model for studying human diseases, allowing researchers to understand disease mechanisms and one of the things the potential treatment is the disease model. Similarly, transplantation, like animal biotechnology, can be explored for the possibility of using animal organs for transplantation into humans. Already people have tried out many such transplantation, potentially addressing the critical shortage of the human organs. Of course, a lot has been done in your institute also, vaccine development, where animal cell cultures are essential for producing various vaccines. Potentially addressing the critical shortage. So these are kind of biotechnology for the human health. I thought it will be worthwhile just cell culture. Not that I understand many of these things, but I collected some material as to what are the emerging research areas in technology human health. A few of them I can highlight, may not be new to you, but animal genomics and multi-omics platforms, technologies which are being researched are whole genome sequencing, SNP arrays, RNA sequencing, and epigenomics. The applications are breed improvement through genomic selection indices, identification of climate resilience and disease resistance alleles, and multi-trait selection models using integrated genotype databases. So emerging research area is pan-genome construction for bovine breeds, omics-based fertility mammals. This is one area which I found reported in the literature. Microbiome engineering and functional feed additives, where technologies are metagenomics, microbial consortia modulation, and phage therapy, where basically we are looking at methane reduction through human microbiome editing and improve nutrient absorption, gut health, and disease resilience, targeted probiotic and prebiotic delivery systems. The research will lead to custom human probiotic libraries, bacteriophage, gut health, and cocktails against mastitis and pathogens. The research similarly, other areas are smart biosensors and AI integrated diagnostics. I heard about this while we were talking with Dr. Tarun on the use of AI and ML in the system. Similarly, so wearable biosensors are coming common. Lateral flow assays, artificial intelligence and machine learning based disease prediction models system with applications early detection of diseases, real-time physiological monitoring, and data-driven decision support for vets and paravets. So emerging research is paper-based with applications, diagnostics, and a smartphone linked multiplex sensor arrays that are coming in. Digital twin models and precision livestock farming, PLF as they call it. IoT cloud computing is the technology, digital twin simulation technology. Digital twin we are using everywhere, so why not here? Digital twin predictive analytics is very important using that as they call it. So it can lead to real-time herd modeling and feed health and reproduction optimization. Climate event stress modeling and adaptive management. These are the applications. Research areas that are emerging are integration of genomics plus IoT plus microclimate data into AI-driven livestock management platforms. These are the applications. The last one is the livestock immunogenomics and mRNA vaccine platforms, where technologies like reverse vaccinology, mRNA synthesis platforms, differentiating infected from vaccinated animals, designs and applications. There are rapid development of vaccines for emerging livestock diseases. Precision immunization strategies tailored by genotype and cross species with zoonotic spillover prevention. The research areas are India-focused vaccine development for PPR, LSD, and Brucella genotype and cross species with zoonotic. What are the challenges? While these researches are being done, I'm sure you have lots of challenges. Some of the challenges I thought I can highlight. What is the scientific uncertainty? When you do anything, you have challenges. Already we are suffering from this problem of scientific uncertainty in the some of the gem crops and gem agriculture and genetically modified crops. You know what is the kind of dialogue that goes on. So theoretical possibilities of unintended consequences. People always talk about unintended consequences. Gem crops and uncertainty about the long-term effect. We don't know, sir, it is okay today, but we don't know what is going to happen tomorrow. So theoretical regulations differ from country to country, and as a result, you find sometimes difficult to operate across. Difficulty in harmonization, funding issues, IP protection issues, biopiracy concerns. I don't know whether you have faced any time some biopiracy to operate across. I'm sure difficulty in harmonization, environmental and societal concerns, environmental impact, and the public perception and acceptance. I don't know how you have the public to take it after you research it. I'm sure many times these modified things are not accepted by the public for fear of various things. So we have to worry on that. And food sovereignty. I don't know how you have. Capacity is a major problem. Institutional capacity, need for expertise and capacity. Not very happy that you also have students who are doing you. So we have to PhD. I think this is the one way you are able to increase the capacity of researchers in the country. Need for robust monitoring and surveillance, that's again capacity. What are the solutions? The solutions are we should increase funding and investment. More investment in research, infrastructure and training is crucial for advancement of animal diet. Again capacity. We need to build capacity and develop training programs and partnerships to build human capacity is essential. International collaboration. Certainly it helps. We need to build. While I was trying to dissuade Professor Murthy from doing international collaboration, but that is only for funding part. International collaboration are always open. Certainly it helps. Yes, funds are welcome, but technology should come and we should fill our gaps with that as far as the technology is concerned. So collaboration and knowledge sharing between countries can help to overcome the challenges and accelerate progress in animal biology. But technology should come. Public engagement and education, raising public awareness and addressing concerns. Collaboration and knowledge sharing between countries is important for fostering acceptance and accelerate progress in animal biology. Your outreach program, public engagement. I think it's going to help in that way when you will be able to take your research, your ideas into the public domain. Strengthening regulatory frameworks, developing clear, consistent, and transparent transparent regulations is necessary to ensure responsible development and use of animal life in the public domain. Strengthening regulatory frameworks. One of the areas which everybody works today is the sustainability development. To ensure responsible development, that science what we are doing, how it is actually helping the SDGs. So when I tried to map, I found there are very good things which animal biotechnology is doing. SDG 13, which is a climate action. That science what heat resistant breeds you are developing, methane reducing feed you are making. So that's the impact on SDG 13. Similarly, SDG 2, zero hunger and genetic gains, there is disease resistant breeds which you are talking of. So that leads to the SDG 2. Similarly, you take SDG 9, which is industry, innovation and infrastructure. And you are setting up biotech startups today. I'm very happy Dr. Hadan was talking that he has got a program which he was talking about. So biotech startups, R&D infrastructure of course is very important. SDG 15, which is life on land. So genomic improvement, which is leading to that, and land pressure reduction. The SDG 3 is the good health and well-being. You are already doing the disease modeling and the vaccine development which comes in the way of good health and well-being. SDG 12 is responsible consumption and production, which you are managing with the genomics and gut micro work. That is what we are doing. Now, country has launched World Health is responsible. What is called One Health, One Nation mission. So, animal genomics, India, One Health, One Nation mission becoming integral. A holistic approach, now integrating human health, animal health, and environment is called One Health, which has been endorsed by WHO and adopted by India in the National One Health mission. A holistic approach, I think it was in 2022, human health. Animal health. You can see from there, environment, animal, human, all of them put together with the well-being of population being taken care of, health of animals will be taken care of. I think it was. But how the animal biotechnology is contributing to that is also highlighted. For example, animal health, all of them put together. The vaccines, well-being of diagnostics and CRISPR-based disease resistance, reduce outbreaks and antibiotic technologies. Other contribution in the human health is zoonotic health, disease surveillance and disease modeling, like swine flu, avian flu, and all that. Disease resistance, reduce outbreaks. Xenotransplantation and organoids for biomedical applications. This is another biotechnology. Disease surveillance, environmental health. We are all talking of low emission livestock. Better manure, xenotransplantation, and feed innovations, reduce environmental pressure. Public health preparedness, rapid response platform for emerging disease from animals. Better manure, that is what the contribution. And feed innovation. Now, what is the value creation by animal biotech? Public health preparedness. After all, when you do what, something has to be coming out in terms of value. Better manure. The economic contribution, if you look at feed innovation. So, the value creation, it contributes about 4.1% of GDP and 25.6% of the total agriculture. The economic contribution, that is what the animal biotechnology. So, employment generation provides employment to 8.8% of the population. 25.6%. Income generation, 16% contributes to the income of small farm households. Employment generation to the rural. Provides employment to 8.8%. The nutritional security provides protein-rich income sources, enhancing health and nutritional well-being. Soil fertility is another gain. Livestock manure use and nutritional security and boosting crop yields and promoting sustainable farming. Nutritional well-being. It is an asset for fertility all the animals. Livestock acts as a collateral for credit and investment, helping farmers finance agricultural activities. So these are some of the value chain what you are able to gain out of all the animals. Livestock acts as a how do you figure as far as the scenario in terms of growth is concerned? Global scenario of the value chain, the animal biotechnology global market report 2025 says that the growth is at the CAGR of 10.2%. And expected market size is about 48.68 billion in 2029. This is a global scenario. That India, we have 120 plus million rural households derive partial or full income from dairy, poultry, small ruminants or mixed global systems. India contributes about 4.3% to the GDP and 15% of agriculture exports, yet receives only a fraction of ruminants or mixed global systems. This is what I wanted to highlight. India's dairy sector is the largest in the world by volume, but trails in yield per animal, disease control and sustainability. We have one problem. In terms of market value, we are not bad. 10.11 billion in 2022 and expected to be about 12.45 billion. That is, we have one kind of a goal we have set for ourselves. We are not bad. 10.1. So we all know that this is the era of Viksit Bharat. In animal biotechnology, India of course is looking at Viksit Bharat with bioeconomic growth, Atmanirbharta, jobs and entrepreneurship, health secure India, and so on. Biotechnology. So animal biotechnology will play a pivotal role in achieving a sustainable growth, food security, and health and future generation, health secure India, continued innovation and integration, animal biotechnology, national priorities through that the sector is poised to shape a resilient and prosperous future for India's agriculture and allied sectors. Continued innovation. So what is the way forward? Where do we go from here? So considerations regarding adoption of biotech should build upon existing conventional technologies. Continued innovation. We should be integrated within the framework of a national livestock development. So should be technically, economically, and socially affordable. Should build upon existing policy and institutional considerations should be adopted. We should be integrated within the framework biosafety regulations, and we should also take care of technically, economically, socially affordable. Should build upon existing policy. We should also look at capacity building. We should be actively involved in livestock biotechnology research with well-equipped laboratories and qualified human resources. I think NIAB is shining at that. We should also look at capacity. Started in a small way, but it is growing in that sense. So I think we should have many more and much bigger organizations which are dealing with the research in this area. I think important that the developed capacity maintains a strong base of applied and adaptive research and some level of training to keep abreast with the latest developments. This should happen in this area. I think important to conclude, I would say advances in biotechnology have evolved at a very fast rate in some level of training to keep abreast. Breakthroughs in farm animal genome sequencing, molecular marker technology advances, and other biotechnology applications will provide new possibilities for research to improve and modernize the livestock industry. In the future, animal genetic composition will be available, allowing for the prediction of animals' production capacity before birth or even as a fetus, as well as selection of animals best suited to certain production settings. Allowing for animals to live, it is critical that necessary knowledge before birth is handed down through generations, as well as selection of animals. If population's genetic information has enough diversity, it is more likely to adjust successfully to environmental changes. Agriculture, medicine, and environmental conservation are all being revolutionized by animal biotechnology by using methods such as cloning, CRISPR-Cas9, gene editing, and developing transgenic organisms that have produced disease-resistant livestock, increasing agriculture productivity and protecting endangered species. So generally, or genetically, I would say, modified animals are used to produce human-compatible organs and biopharmaceuticals, which address shortages in healthcare and improve. So generally, If we can achieve that, I think we will be able to get somewhere as far as this requirement is concerned. Stem cell research provides new treatments for various conditions, and selective breeding encourages sustainability and food security. Somewhere as far as this obstacles to progress include ethical considerations, regulatory complexity, safety concerns, and public perception. In conclusion, while the prospects of biotechnology are vast and transformative, addressing the ethical, regulatory, safety, and public perception challenges will be crucial for its sustainable and responsible advancement. We need to work together to overcome all of them and take this particular tool available to us for the human benefit through our continued research and expansion and capacity building. Thank you very much. Wish you once again all the best.
Thank you, sir. Thank you for that brilliant lecture. If you may permit, we can have some questions from the audience.
Thank you very much. Wish you once again all the best.
Thank you, sir. Thank you for that brilliant lecture. If you may permit, we can have some questions from the audience.
Thank you very much. Wish you once again all the best.
Thank you, sir. Thank you for that brilliant lecture. If you may permit, we can have some questions from the audience.
Thank you very much. Wish you once again all the best.
Yes, sir.
Thank you, sir. Thank you for that brilliant lecture.
Hello, sir.
If you may permit, we can have some questions from the audience.
Thank you very much. Hello.
Yes, sir.
Thank you, sir. Thank you for that brilliant lecture.
Hello, sir.
If you may permit, we can have some questions from the audience.
Thank you very much. Hello. No.
Yes, sir.
Thank you, sir. Thank you for that brilliant lecture.
Hello, sir.
If you may permit, we can have some questions from the audience.
Hello, sir. So it's a very interesting talk. So my question is regarding some of the policies that the government is, it might want to introduce or something. So like how we saw in the case of this lace, so it's a very interesting talk. So my question is regarding some of the policies that there is a lot of controversy going if it is ethical or if it should be permitted or not. So like how we saw, especially when IP comes in relation to IP of whatever we. My question is regarding in the future. So if there is a lot of patents or somewhere, if it is ethical or if we should use it in India, and that's how we saw especially when IP comes. So how should the like what sort of policy can the government do so that we can retain talent here but still encourage them to go off? So especially when IP comes.
Yeah, I think your question is complex. First thing is there are two issues. One is acceptance of anything which is genetically modified and which leads to certain changes, but yeah, in terms of productivity, in terms of health, in terms of resilience, is better. All of us know. Acceptance. Now, we are also worried whether there are any adverse effects of that genetic modification. Yeah, in terms of why the population is worried. And this is happening in the case of agriculture. Genetically modified, all of this is what is happening. Now, while people are modified, they have got gene sequences that are used for modification. They have even taken a patent for that, but they are not able to commercialize it. That is where the policy comes. While people are India took a decision that High Court decided that we will not do. They have even taken a patent for that. So same thing will happen here. My answer to that is where the I think we are not doing a holistic approach. That we will not. Holistic approach is you should demonstrate. So same thing will happen. That there are no adverse impact of that money. I think we are, for example, when the cotton genetically modified cotton came. Holistic approach. Then everybody thought that you should demonstrate modification we are doing. It has got a problem with respect to antibiotic resistance. For example, and as a result, they said adjacent crop is also going to be affected. That was a fear. Now, scientists should have done moral exercises by which they should have seen what is the impact on the adjacent crop, how much is the antibiotic resistance is developing. I just gave an example of crop because I know about it. Other thing I don't know. Moral exercises. So you should do a similar thing whenever you are doing this kind of work in your domain. Coming to just an example of crop, if the commercialization does not take place and the scientist does this kind of a work, and in India we don't permit him to use it for commercial purpose, he will get the same. And he will certainly have a problem of retaining it. He will go to a country or a place where his idea is better received. The only answer for that is use it as early as possible, get into regulations. That's why I talk so much about that. Main thing is get into regulations or a place where create his idea, a ecosystem among scientists to convince the public, create the public perception which is positive, and that is our job. Outreach programs are one among them. Here we have to tell people that this is not, for example, farmers. Many things which the scientists do in agriculture, farmers don't accept, and that is our job. For example, we talked about nano urea. Now nano urea is not being taken by the farmers. All of us know that nano urea is better in terms of its application. For example, farmers are not. We talked about nano urea because we have applied that science in the Kendra. We have done all that study, but we have not actually interacted with the farmers, demonstrated to them, gone to the field, shown them the whole thing. So there is a disconnect with the user. Same top thing happen in your field also, that the dairy people will suffer and demonstrated to them. So that's the only way we can retain people. So there is a disconnect with the user. Same top thing happen in your field also, that the dairy people will suffer and demonstrated to them. So that's the only way we can retain people. So there is a disconnect.
It's an excellent talk, sir. So, in one of your slides, you've spoken about biotechnology's role in geopolitical stand. So, we know that the missiles that you, it's an excellent talk, sir. So, in one of your slides,
I didn't put any politics.
Biotechnology's role in no, like my question is, we know that the missiles capacities, defense and geopolitical stand. So, what do you think about the role of biotechnology in a nation like my geopolitical stand in the world?
I come from defense. I have
defense and geopolitical stand.
Biological warfare.
The role of biotechnology
is a major fear.
in a nation like
All these microbes, all these things what they do can create major problems. The viruses and all those things. Biological warfare.
the role of biotechnology.
So the major fear. As far as the geopolitics is concerned, all these microbes, the world has decided that chemical and biological warfare will not be done. The viruses and all. Treaties among the countries who have said that no biological will not use chemical. The world will not use explosive warfare. So this is the geopolitical position. But despite that, there are a large number of countries who are creating a bank of bio weapons. But people also say that COVID was one bio. There are a large number of countries. I don't know to what extent it is correct, but there are lots of proofs which have come out that it was an exercise which was being done by China to develop a bioweapon. Which came out of their laboratory by accident or otherwise, and finally we all had to suffer. So, an exercise, the work is going on. I am also saying that no country should remain oblivion and live in the comfort that we have signed a treaty, so we don't need to worry. The work is going on. I am not talking of offensive technology, but I am talking of defensive technology. If tomorrow there is a bioweapon comes against us, and hence you should know what's the bioweapon, then only you can identify a defense against it. So you have to work in that. If tomorrow, like that we were doing in the chemical weapons, so hence we should know what's the. We were even storing some chemicals which used to be used, and finally when we signed the treaty, we finished all, we closed all of them. But what we did, we kept the formulations with us. We were all the formulations are kept in a bank, sealed, used to be covered. So if I need to use it tomorrow, I'll be able to use all of them. So geopolitical is a very sensitive subject with us. Biology, bio weapons or bio things to be developed if I need anything which is bad for the human. So geopolitical is a very sensitive subject with us. Biology, bio weapons.
That was fantastic talk. I have a different kind of perspective. So we are training a lot of grad students, like they finish their PhD, graduate students.
Graduate students.
Yeah, they finish their PhDs. All over India, we are training a lot. After they finish their PhDs, right away they will leave for their postdocs and graduate students. Take it to Europe or US or any other place. All over India, we are training. The worst way we are training them to the end, and ultimately we are sending them somewhere outside India. So, what are your suggestions to keep them back with us? And what are your suggestions basically to keep everybody and all the science? Yes, exactly.
This is one of the major problems our country has faced and facing.
What are your
Best of our students, whether they are from your institution or other one of the major problems, tend to go abroad for and face the PhD or postdoc. And many of them students, most of them don't. Whether they are from your institution. The only way to sustain them in our country is to go abroad for by two things. The PhD or postdoc. Number one, we should give them the same kind of challenging research and the only way which exists outside. In our country, if we don't give, like I was talking, number one, if KFR has to be like one of the Cambridge labs or the Harvard labs, whatever in our country, we need to have the same kind of thing. We can't do it only with 300 crores. If KFR has to be, we need to expand the Cambridge lab. So unless you give them the same ecosystem, where they don't have to, we need to have the same those instruments and equipments and things like that with the use. Second thing is, unless you give them the same ecosystem, research quality also has to be shown to be done. And one of the major things which I am noticing is, we don't have a postdoc culture in our country. Research, most of the postdocs is done outside. So we have to create the postdoc culture. The postdoc can be done only if the faculty has a very active research program within the labs. So we have to. I'm not talking of national labs. I'm talking of academic. The postdoc from where the ICERs. Why did we create ICER? We created ICER so that in the field of, I'm not talking of pure sciences, chemistry, physics, biology, mathematics, we are able to get the best talent which will be used for research and education in the universities. That was the idea. But today, after doing MSc, most of the students from ICER go abroad for PhD. They don't even do PhD there, which will be used for. Why don't they do PhD there and education in the universities? That was the idea. Job opportunity, second thing, the ecosystem. ISL ecosystem, even today, despite that much of investment, does not compare equal to the ecosystem in Germany or Japan or whatever. Second thing, so that is a thing why people go. ISL ecosystem. Second thing is, the faculty research does not compare, is not of the same class as they find in MIT or something. So that's why they go for postdoc there. Second thing is, third one is branding. Faculty research. I'm from MIT. I'm sure all of you, when you write in your CV, you write MIT, PhD, or postdoc, or Harvard, right? Why do you write branding? Nobody writes, I'm from Central University, has a CV, you write MIT, PhD. So that is the kind. These are the reasons. To retain them, these three things happen. Why do you write branding? You have to create a strong postdoc culture from Central University and strong ecosystem for this. We cannot live with this kind of substandard research facility. I'm happy that you are growing your area, you are building equipment, you are building things. And you were also telling about TIFR, new TIFR. I mean, that's the way we have to go. Then only they will. I'm happy that you are growing your area. Challenge, sir, challenge. And you were also. I didn't go abroad. TIFR, new TIFR. I mean, that's the way we have to go.
Yeah, thank you very much.
Neither for study nor for learning any technology. Challenge, sir, challenge.
Thank you so much, sir, for your wise
I didn't
words.
go abroad.
We are now request Dr. Sharma ma'am for. Yeah, thank you very much.
Neither for study nor for learning any technology. Challenge, sir, challenge.
Thank you so much, sir, for your wise
I didn't
words.
go abroad.
We are now request Dr. Sharma ma'am for. Thank you very much.
Neither for study nor for learning any technology. Challenge, sir, challenge. I didn't tell you anything new.
We are now request. Thank you very much.
Neither for study nor for learning any technology. Challenge, sir, challenge. I didn't tell you anything new.
We are now request. Thank you very much.
Neither for study nor for. हाँ बिल्कुल खोलिए. I didn't tell you anything new. हाँ बिल्कुल खोलिए.
That's our even management team, Dr. Prasad.
हाँ बिल्कुल खोलिए. ये भी है.
That's our even management team, Dr. Prasad. Like to not build like this.
ये भी है. No. I will request the following. Let it all remain here. I'll get it collected and take it to Hyderabad home. Rather than taking it to Delhi.
Like to not build like this.
ये भी है. No. I will request the following. Let it all remain here. Thank you. I'll get it collected and take it to Hyderabad home. Rather than taking it to Delhi.
Like to not build like this.
ये भी है. No. I will request the following. Let it all remain here. Thank you. Get it collected and take it to Hyderabad.
Thank you, sir. Thank you,
Rather
ma'am.
than taking it to Delhi.
So on behalf of NIB Event Organizing Committee, I extend my heartfelt gratitude to each one of you for joining us on this important occasion, the 14th Foundation Day of National Institute of Animal Biotechnology. So on behalf of NIB, built on a vision, strengthened by the people and driven by purpose. Today, as we celebrate the milestone, we are reminded of the dedication and collective efforts that have shaped NIB's journey so far. Built on a vision, first and foremost, I extend my heartfelt gratitude to Dr. Vijay Kumar Saraswat for gracing us with his presence and delivering an inspiring and enlightening lecture. Sir, your words have truly inspired us and added immense value to this special occasion. A special thanks to all the others present for their presence. I would also like to sincerely thank our director, ma'am, for her unwavering support and encouragement, which have been instrumental in making this event a reality. A special word of appreciation goes to the organizing committee. I also wish to acknowledge the contributions of all faculty members, administrative staff, finance, security, and supporting staff, and everyone who has worked behind the scene to make this. I also wish to acknowledge today a success. To all our distinguished guests and attendees, thank you for joining us and making the Foundation Day celebration even more meaningful with your presence. I now request you to please join for a group photograph, followed by guests and attendees. Please come joining us for making the Foundation Day celebration even more meaningful with your presence. I now request you to please join for a group photograph, followed by guests and attendees. Please come joining us for making the Foundation Day celebration even more meaningful with your presence. I now request you to please join for a group photograph, followed by guests and attendees. Please come joining us for making the Foundation Day celebration even more meaningful with your presence. I now request you to please join for a group photograph, followed by guests and attendees. Please come joining us for making the celebration even more meaningful with your presence. I now request you to please join for a group photograph, followed by please come join us for the I now request you to please join for a group photograph, followed by please come join us for the I now request you to please join for a group photograph, followed by
you. Thank you very much. Thank you. Thank you very much. Thank you. Thank you very much. Thank you. Thank you very much.